Well, hello, ladies and gents. Here we are in the last segment looking at bulk transport, where cells learn how to drive cars. Let's see. So sometimes solutes are really big, too big for just a channel or a carrier protein to be able to translocate the substance into or out of the cell. Or you're trying to consume too much material to have it flow through one tiny little channel. So what happens is bulk transport. Now, this is something that does require ATP. It's a type of active transport because what it does is it actually rearranges the cytoskeleton. What do you think builds the proteins of the cytoskeleton? Yes, enzymes. Okay, so enzymes can build up the cytoskeleton. But if you have enzymes that can build the cytoskeleton, that means there are probably also enzymes that can deconstruct the cytoskeleton. So, what some cells have the capacity to do is actually break down their cytoskeleton and rearrange it in different ways to change the shape of the cell and actually swallow up materials from outside the cell, or they can take their vesicles and fuse them and dump their materials outside of the cell. So it's really spectacular what cells sometimes have the capacity to do. So let's first look at the idea of exocytosis. Exo means outside of, cyto is cell, osis means condition of. So exocytosis is where materials produced inside of the cell have membranes fuse with the plasma membrane and dump the contents outside of the cell. So remember with endomembrane transport, where the Golgi can modify and activate some proteins and then ship them off to where they need to go? Well, if those membranes fuse with the plasma membrane, it can open up and dump those contents out. So maybe a cell is producing insulin to help lower your blood sugar. Well, that insulin needs to travel through your bloodstream. So the vesicle carrying the insulin will fuse with the plasma membrane, open up, and dump the insulin out. Okay. Endocytosis is sort of the reverse process. With endocytosis, solutes can be swallowed up when the plasma membrane wraps around the substance. So here you see sort of the red cell. It'll wrap around and form a vesicle around the green solute. I'm sorry if you have red-green color blindness. It's the gray oval surrounding the gray rectangle. So what happens is the cell membrane will again actually wrap around and form a membrane around that, and it's basically packaged in a vesicle. Now that can happen with something like a macrophage. Macrophages are part of our immune response, so your non-specific immunity. What they'll do is they'll go and swallow up things like pathogens, like bacteria. And then what's cool is that vesicle will then fuse with your lysosomes, and then the lysosomes release their contents and destroy the solute or destroy the pathogen. Penocytosis, peno means to drink. So penocytosis is cell drinking. So if cells sort of swallow up a bunch of water, that's sort of how sponges eat. Sponges are animals. They're the first animals, really simple animals. And what some sponges do is they have cells that will swallow up seawater. And in the seawater is detritus. It's basically little bits of plants and animals that are breaking down. So they can swallow the water and grab that detritus that's floating around the water. And then they'll break that down and consume that. And that's how sponges eat. Phagocytosis, phago means to eat. So that's cell eating. Again, you can swallow up those particles. That's like one of your macrophages eating a bacterial cell. And then there's what's called receptor-mediated endocytosis. Receptor-mediated just means that a solute needs to dock on the receptor, and that can pull in those contents. So here you can see the solute, LDL, maybe this is a type of low-density lipoprotein. The solute can dock there, get caught up with the vesicle, and then it can fuse with the lysosome and get destroyed. Well, this is just like a little visual review. If you get the actual lecture, I can try to post the actual lecture. This is just a little 3D thing that I drew last year as like a review. So, we can zoom in and look. Again, if it's small and nonpolar, it can diffuse right across the membrane in between the phospholipids. So there's a nice example of diffusion, a type of passive transport. If it's large, charged, or polar, it can move through a channel protein, which there you can see the channel, or you can have a carrier protein, that'll change its shape. Okay, now if you have active transport, there's active transport. Active transport uses a carrier protein, but notice solutes go from areas of lower concentration to areas of higher concentration. And what you need is ATP to cause that shape change. Here, if it's something large, charged, and polar, if it's like a meteorite, that's not going to squeeze between those phospholipids. All right, well, there you have it. If you have questions about stuff, please don't hesitate to let me know. But otherwise, there's chapter 7.